as well as some of the general functions of the 3 muscle types in your body. And towards the end of today's lecture, it will cover one of your potential essay questions. Okay, so muscles. All muscle cells, they specialize in contraction. And when we say something contracts, it means it just decreases in length, or it shortens. So if something contracts, it's going to decrease in length. And that contraction will cause movement, whether it's of your skeleton, of your heart, or different smooth muscles within your internal organs. So we have 3 types of muscles. Skeletal muscles, which are mostly attached to your bones. They can also be attached to your skin. We have cardiac muscle, which is exclusively found in your heart. And then smooth muscle is found in a variety of organs, such as your digestive tract, the uterus, your eye, blood vessels, etc. So for the next handful of slides, we'll explore these 3 muscle types in a little bit more detail. Starting with your skeletal muscles. For the most part, again, they're going to be attached to your bones via connective tissues called tendons. In addition, skeletal muscles can attach to the skin of your face to allow for facial expressions and the movement of your face. And in addition, you can find skeletal muscles in some internal organs that will allow for the movement of fluids. If you look at skeletal muscles underneath the light microscope, it looks something like this. Skeletal muscles, they are these long cylindrical fibers. So you'll see that they're arranged into cylinders. And a unique feature is they are multinucleate, meaning they have multiple nuclei hanging out along the periphery of the muscle fiber. Also, when you look at it underneath the light microscope, you'll see that it has this stripe-like appearance, and we say they are striated, and that's due to the arrangement of proteins within inside the muscle fiber. And with all these cylindrical muscle fiber cells, we're going to arrange them into bundles, as we see here. Skeletal muscles allow for voluntary movement. That's movement we have conscious control over. And in addition, skeletal muscles are involved in somatic reflexes, such as the knee-jerk reflex. So what are some functions? Well, when muscles are attached to your bones via tendons, and the muscles contract or relax, it allows for the movement of your body and skeleton. When they're attached to the skin of your face, they allow for hundreds of different facial expressions. With muscles in our chest and in our back, it can support our body and allow for posture. Muscles around our joints help stabilize them and keep them in place and prevent them from being dislocated. One big diaphragm we have in our body is one big muscle we have in our body is the diaphragm, which contracts and relaxes to allow for breathing. And when we're cold, our skeletal muscles contract rapidly to cause shivering. And then skeletal muscles can also be used to allow the movement of fluid throughout your body. Or for example, there are skeletal muscles that control the excretion of fluid urine or feces. And lastly, skeletal muscles can add some protection and padding to pad our internal organs. So skeletal muscles have a variety of functions. They're not just used for the movement of your body. Going on to our second muscle type, we have cardiac muscle, which is only located in your heart, and specifically it makes up the walls of your heart. If you look at cardiac muscle underneath the light microscope, it would look something like this, where each cell is branched in structure. And unlike the skeletal muscle fibers, these cells just have one nucleus within them. And when you look at them underneath the light microscope, you'll also see stripes within them due to the arrangement of proteins inside, where you'll have these darker regions and lighter regions. Another feature of cardiac muscle are these cells are physically connected with these channel-like structures referred to as gap junctions. And that allows a signal from one cardiac muscle cell to simply and quickly travel to the next one. And this will allow for coordinated contractions of your heart. You can also say the cytoplasms are continuous with each other. With our heart muscle, we know that we do not have voluntary control of their contractions and of our heart rate, and thus cardiac muscle is regulated by your autonomic nervous system. Because they're only located in your heart, when they contract and relax, it allows you to pump blood and also to regulate blood pressure. If you increase your heart rate, that will relate to an increase in blood pressure and vice versa. Smooth muscles, they're going to be located in various internal organs, and based on the organ it's located in will determine its function. So for example, in females, there's smooth muscles in the uterus, and their contractions help with child labor. In the alimentary canal or the digestive tract, there's smooth muscles that can contract to allow for the mechanical digestion of food and also the movement of food through that tube. On the first day of class, we said that there's smooth muscles within your blood vessels, which can contract or relax to allow for vasoconstriction or vasodilation. That would regulate body temperature as well as blood pressure. Within the bronchioles or the airways of your respiratory system, we have smooth muscles that can contract or relax to allow for bronchodilation or bronchoconstriction, which we described on Tuesday. Also, when we described vision, we said we have smooth muscles in the iris of our eye, which controls the diameter of your pupil, or how much light enters your eye. And then lastly, you'll find smooth muscles within your urinary bladder, and when they contract, it can help with urination. So we have smooth muscles distributed into different organs of the body, and they have a variety of different functions. When you look at the cells underneath the light microscope, they have this spindle shape. Like cardiac muscle, they have a single nucleus, and they're called smooth muscles because they have no striations within them. And visually here, you can see that the smooth muscles, they arrange into sheets. So these smooth muscles, they're showing you that it belongs to the iris of your eye, regulating the diameter of your pupil. And these smooth muscles, they're showing you it's part of your digestive tract to allow for the digestion and movement of food through. For all the smooth muscles, we do not have voluntary control of their contractions or relaxations, and thus it's regulated by your autonomic nervous system. So those are your three muscle types: skeletal muscle, smooth muscle, and cardiac muscle. You'll look a little bit more of these three muscle types in lab. But for the rest of today and this lecture, we're just going to focus on the skeletal muscles. We'll look at their anatomy a little more in depth, and then we'll describe their physiology to allow for muscle contraction and muscle relaxation. So, skeletal muscles, they control voluntary actions of your body, and thus they're a part of your somatic nervous system. And if we look at this whole entire muscle organ, maybe let's just say this is your biceps muscle, for example, it's divided into many layers. So this outer wrapping is known as epimysium. It's a connective tissue that wraps the entire muscle organ. And then you'll notice the whole entire muscle is then divided into these fascicles. So there's a fascicle circled in blue. This would represent another fascicle. Let me grab blue. Here's another fascicle. Here's another fascicle. All these bundles are fascicles that make up the muscle organ. And each fascicle is wrapped in another layer of connective tissue known as perimysium. And then within each fascicle, now it's subdivided into even more cylinders. These cylinders would be your myofibrils, aka your muscle fibers. So in green, those are your muscle fibers. And they're wrapped in another layer of connective tissue known as endomysium. Muscle fibers surrounded by endomysium. And then lastly, you have one more level of cylinders, which is your myofibrils, made up of various proteins. So all three of these connective tissue layers, endomysium, perimysium, epimysium, they go through the entire muscle organ, and they all connect together to eventually form the tendon, which connects your muscle to your bone. So we said on the first day of class that the cell is the basic structural and functional unit of life. So thus the cell, or the muscle fiber, will be the basic structural and functional unit of the muscular system. So let's look at the cell in a little bit more detail and identify its parts. So this whole structure right here represents a single muscle fiber, also known as a muscle cell. Here is a single muscle fiber as well. This would be another muscle fiber, and another muscle fiber, and another muscle fiber. Okay, so when we talk about the structures of the muscle fiber or the muscle cell, we give certain structures unique names. So the sarcolemma of muscle fibers represents the plasma membrane of it. So this would also be the sarcolemma, or the plasma membrane of the muscle fiber. So sarcolemma is a general term. There's two more specific terms that describe specific regions of the sarcolemma, or specific regions of the plasma membrane. First is the molar end plate. The molar end plate of the sarcolemma is the part of the plasma membrane that will be in communication with the axon terminals of a neuron. And then transverse tubules, or T-tubules, it's the part of the sarcolemma that invaginates inwards. And these T-tubules will eventually carry the action potentials deep into the muscle fiber. Another term you should be familiar with is sarcoplasma. What does that sound like if we just talk about cells in general? Sarcoplasm sounds like cytoplasm. So thus it's the cytoplasm of the muscle. So on your notes you can write equals cytoplasm if that helps you. And we know that cytoplasms of cells or sarcoplasms of muscles, they're going to contain various cellular organelles. One important organelle of muscle fibers are mitochondria. Mitochondria produce a ton of ATP, and to give you a little preview, muscle contraction requires a lot of ATP, and thus muscles need a lot of mitochondria to make that ATP so that the muscles can function. In addition, you'll see later on that ATP is also required for your muscles to relax. In the sarcoplasm, you'll also find glycogen hanging out, and that could be an energy source for the muscle. And you'll also find myoglobin, which stores oxygen. So if you remember, in aerobic cellular respiration, to make ATP, we take some type of nutrient, we use oxygen to make carbon dioxide, water, and most importantly, ATP. So this ATP, when we talk about the muscular system, it will power muscle contraction and relaxation. Now for this oxygen, some of it, not all of it, but some of it comes from that myoglobin. And some of the nutrients to make the ATP comes from the glycogen. Glycogen can be hydrolyzed or broken down into glucose. And then glucose can then enter aerobic cellular respiration to help you make ATP. In addition, fatty acids, they can act as a nutrient source for your muscles to make energy. And then we can also get oxygen from hemoglobin. So those are the nutrient sources that we use to make ATP to power muscle contraction and muscle relaxation. Another organelle of the muscle is the sarcoplasmic reticulum, meaning smooth endoplasmic reticulum. That's what the sarcoplasmic reticulum is. It's the smooth endoplasmic reticulum of the muscle. And the job in the muscle is to store calcium. Ca2+, calcium ions. So you'll also see that calcium ions are also absolutely required for muscle contraction, in addition to ATP. We said already that your muscle fibers of skeletal muscles are multinucleate, meaning they have multiple nuclei. You can see a few of them here in purple. And they're striated due to the arrangement of proteins within them. You can see we have these darker bands, lighter bands, darker bands, lighter bands. That allows for the striations. And when you look at the whole entire muscle fiber or cell, it's divided into these other cylinders known as myofibrils. highlighted in red is a single myofibril. But you can see there's many other myofibrils that make up the muscle fiber. So the next thing we're going to do is look at the anatomy of the myofibrils a little more closely. This is where most of the work happens to allow for muscle contraction and muscle relaxation. Okay, so we're going to pull one of these myofibrils out, which is seen right here, and we're going to look at its anatomy or structure. You can see that the myofibril is divided into repeating subunits known as sarcomeres. So here's a sarcomere, here's another sarcomere. And sarcomeres will span the entire length of the myofibril from beginning to end. If we take one of these sarcomeres and we look at its molecular structure even more closely, we can see the proteins that make it up. So what makes up the sarcomere? These proteins known as myofilaments. And there's three types of myofilaments. We have thick filaments, which are conveniently thicker in structure. We have thin filaments, which are thinner. And we have elastic filaments, which act as molecular springs. Those are these guys that look like springs. On the next slide, we're going to look at these three myofilaments a little bit more closely. Let's start with the thick filaments. You should see that the colors are matching. Red here shows you the red thick filaments in the sarcomere. And if we pull it out, we can see the many thick filaments here. And what makes up the thick filaments are myosin motor proteins. So these proteins are derived from DNA, and they're motor proteins because they help with movement. If you look at a single myosin protein, it has a head and this tail-like structure. In addition, you'll have another component intertwined with it. So this represents a single myosin protein. This is the tail, and these are the heads. One of the heads can bind to the actin protein, which we'll describe next. And the other head can bind to ATP. So thus, myosin, its heads, it each has a binding site. One binds to the actin protein, and the other head binds to ATP. And kind of schematically, we can arrange all the myosin together to make the thick filament. And that's what we see up here. We have these myosin proteins with the tail and heads that can bind actin and ATP. And when you kind of stick them all together, you make the thick filament. Now let's look at the thin filaments in purple. The thin filaments are made up of the actin protein. So a single B represents actin, and actin kind of connects with each other in a necklace-like conformation. And we can have two actin strings kind of intertwined with each other. So at a certain point during muscle contraction, this myosin head with its actin binding site, it can bind to the actin. So here in purple, we see the individual beads of actin kind of intertwined together. That's one component of the actin thin filament. Another thing that makes up the thin filament is troponin. Troponin is in yellow. And you'll see later on it will bind the calcium ion. And troponin is attached to a cable-like protein known as tropomyosin. So troponin is bound to tropomyosin. And the job of the tropomyosin is to cover these binding sites for myosin at certain times. This picture here illustrates that concept a little bit better. Let's look at this diagram here. We have the actin balls. We have the tropomyosin in purple, those cables. And then in pink, we have the troponin. When the troponin binds calcium ions, it moves the tropomyosin cable off these black binding sites for the myosin head. And thus, the myosin head would be able to interact with the actin here. But during muscle relaxation, what happens is the tropomyosin cable, it covers up those black binding sites, thus not allowing for muscle contraction. So right now we're just talking about the anatomy, but later on we'll put all these pieces together and describe how it works to allow for muscle contraction and muscle relaxation. Then we describe the elastic filaments made up of titin protein. Here's the elastic filament, which looks like a spring. It's made up of titin. And it attaches the thick filament to the end of the sarcomere. The function of this spring is to return the muscle back to its resting length when it's stretched. All right. Looking at the sarcomere again, here we have our thick filament in green, our thin filaments in purple, and then the boundaries of the sarcomere are known as the Z discs, also sometimes referred to as the Z lines. So between the Z discs, this represents one sarcomere that contains thin and thick filaments. If we look at a muscle underneath the light microscope, you can see the Z disc here, and thus this would represent one sarcomere. Here's another Z disc, and thus this would represent another sarcomere, and so on and so forth. So let's identify the different regions of the sarcomeres. First is the I band, which is only thin filaments. So here's your thin filament in purple, but if we look at this interval right here, we have thin and thick filaments. If we look at this interval over here, though, we only have thin filaments crossing each other. Same with this side over here, only thin filaments. Here's an I band here. Here's another I band here. They appear lighter when you look at them underneath the light microscope because there's no thick filaments intermixed with them. We also have the A band, which is defined by the length of the thick filaments. So this is the A band. Or if we look at it underneath the microscope, this would be the A band. It's darker because it's thicker in structure. It's more dense. So notice in some stretches of the A band, you'll only have thick filament, and in other stretches you'll have overlapping thick and thin filaments. But the definition of the A band is the length of the whole entire thick filament. The H zone, also referred to as the H band, is where we have only thick filaments. So if we look at this interval, up and down, there's only thick filaments here in green. And thus, that would represent the H band. Here would be your H band, and over here would be your H band. It's a little bit lighter in color because we only have the thick filaments. It's darker adjacent to the H band because we have the overlap of thin and thick filaments. Z discs, Z lines, which we identified here, they again mark the boundaries or ends of each individual sarcomere. And another way of defining the Z discs is they divide the I band. Here's an I band, the Z discs divide them in half. And then lastly, we have the M line. Think of M for middle. The M line exists in the middle of the sarcomere. Or you could say the M line is in the middle of the H band as well. So it's important to know these different bands and zones because they're going to change in length during muscle contraction. So let's describe the sliding filament theory of muscle contraction and cross-bridge cycling. This will tell you how your muscles contract. So during skeletal muscle contraction, your muscle decreases in length. Here's your biceps muscle. When it's relaxed. Here's your biceps muscle when it's contracted. If you look carefully, this biceps muscle is shorter in length than this biceps muscle in its relaxed state. Recall at the beginning of this lecture we said contraction means that cell or that muscle is going to shorten in length. So how do we decrease the length of the biceps muscle during muscle contraction? What happens are the thin filaments, these thin filaments, they're going to slide over the stationary thick filaments. When these thin filaments slide over the stationary thick filaments, it's going to decrease the length of the sarcomeres and the length of the muscle. And to slide the thin filaments over the thick filaments, how that occurs are the myosin heads that kind of ratchet the thin filaments inwards towards the middle. So these myosin heads in orange, they kind of pull the thin filaments towards the middle. They crank them towards the end line. And this will happen in both directions. Ultimately, this will decrease the length of the sarcomere. Here's a relaxed sarcomere, and then here's a contracted sarcomere. We've now decreased the length of the sarcomere. And that's what happens during muscle contraction. By decreasing the length of the sarcomeres, we'll decrease the length of the muscle, and that allows for contraction and movement. So if you just compare these two representative sarcomeres in their relaxed and contracted states, what happens to the I band? What does it look like? Does it increase in size? Does it decrease in size? Or does it stay the same length?
It decreases in size?
It decreases in size. So here's your I band. And notice, when we looked at the contracted sarcomere, it has decreased in length. So above this question, you can say the I band, it decreases in length. What was the definition of the H band? It's the length of what? Thick filament. And the A band, it will actually stay the same length during muscle contraction. The H zone, this region right here, which is thick filament only, what happened to it? Did it stay the same length, decrease in length, or increase in length?
Decreased.
It decreased in length. What happens to the length of the sarcomere during contraction? Does it increase, decrease, or stay the same length? Contrasted. Relaxed versus contracted. What happens to the sarcomere length? It decreases or shrinks. And then you can see the Z lines. The Z lines, they come closer together during muscle contraction. So, to recap, the length of the I band decreases. The A band stays the same length. The H zone will decrease in length, as well as the sarcomere. And the Z lines will come closer together. So these are just two other representations of muscle contraction. Here's a schematic image where you see your thick filaments in green, your thin filaments in purple, your Z discs in blue, and you can see this is the relaxed. We have two relaxed sarcomeres. During contraction, we're going to shorten the length of both of the sarcomeres. And you can see that the I bands decrease in length. The H zones also decrease in length. The A band in green stays the same length, and the Z lines come closer together. Here's another representation of the different bands and zones during muscle contraction. I band decreases in length. H zone decreases in length. A band stays the same length. And then the Z lines, they come closer together, reducing the length of the sarcomere. So what pulls these thin filaments inward? It's the actions of these myosin heads. And we're going to look at that mechanism, and that mechanism is referred to as cross-bridge cycling. So let's draw this out on the iPad, and then later on we can summarize it using this diagram. So what we'll draw is cross-bridge cycling. And in summary, it will use the myosin heads to pull the thin filaments inwards to shorten the sarcomere. So here I'm going to draw one representative sarcomere that exists from Z disc to Z disc. And to stick with the same colors on the slide, red will represent your thin filaments. And orange will represent your thick filaments. So what we're going to do is look at one myosin head and its interaction with the thin filament above it. So here's your active thin filament. And attached to it is going to be the myosin head of the myosin protein. And we said that ATP is involved in muscle contraction. So at this state, the myosin head is going to be bound to a phosphate of ATP as well as ADP of the ATP. This attachment right here is referred to as a cross bridge. When the myosin head attaches to the actin, it binds together and forms a cross bridge. So thus, in summary, we could say myosin head forms a cross bridge with the actin thin filament and ADP and phosphate are bound to the myosin head. Let's draw the next phase. Notice the position I'm drawing the actin thin filament. It's not as close to the end here as it was on this side. So let me just draw the picture and then we can explain it. Okay, so what happens in this next stage is the phosphate pops off the myosin head. And that causes the myosin head to perform the power stroke. And what happens is, with that power stroke, it's going to push this actin thin filament inwards, which is what we wanted. This power stroke is going to push this actin thin filament inwards, which is the goal of muscle contraction. That will help slide those thin filaments inwards and shorten the sarcomere. So this happens at various sites of the whole entire sarcomere. We're just looking at one for simplicity. Next, ADP will be released from the myosin head. So we still have our actin thin filament. It's still attached to the myosin head. But now, ADP has popped off. And this is known as the rigor state. For someone that is in rigor mortis, this is how their myosin heads interact with the actin. They're attached and they're kind of locked into that position. So the question is, how do we detach the myosin head from the actin, or how do we break that cross bridge? We do so by binding a new ATP to the myosin head. So in step number 4, we bind a new or a fresh ATP to the myosin head to break the cross bridge. That says new ATP. And when we bind a new ATP to the myosin head, we break that cross bridge. Next, ATP hydrolysis occurs. And phosphate and ADP attach to the myosin head. notice when ATP hydrolysis occurs, the conformation of the head is different. The head, during ATP hydrolysis, it cocks back. So ATP hydrolysis occurs and the phosphate and ADP attach to the head. And the head cocks back. This is the cocked back head, and we say it's energized. So that's step number 5. You should notice that this last step, it should resemble the very first step. So after we get to step 5, the myosin head will then attach to the actin thin filament. And that's what we had in step number 1. The myosin head attaches to the actin thin filament to form this cross bridge. So it's called cross bridge cycling because this process, steps 1 through 5, they repeat. When you get to step number 5, it brings you back to step number 1 so you can repeat the process. And through this process of cross bridge cycling, it will pull these thin filaments inwards. So this series of steps we showed occurs here, but it can also occur at these heads and these heads, as well as these heads over here. And by performing, the key word is the power stroke, that will pull the thin filaments inwards to then shorten the sarcomere. So important terms to remember are the cross bridge, the action of the power stroke, the rigor state, and for number 5, we can call this the relaxed state. Careful, in the relaxed state, the head contains phosphate and ADP, but it's not attached to the actin. Wrong class. So in the relaxed state, there is no cross bridge. So this you could have at least closed the door.
Thank you.
It's okay. You gotta know the secret. You gotta be strong enough to close the door. So this is not the relaxed state because there is a cross bridge state. So, looking at this sarcomere, the goal is to decrease its length. How do we decrease its length? We use these myosin heads. We perform power strokes to ratchet the thin filaments inwards. This occurs through cross-bridge cycling, which is depicted here. it occurs in a cyclical fashion. These steps, 1 through 5, correspond with the steps on my iPad drawing. So you can use this to help you study. Notice, remember these black circles on the actin thin filaments? That's where the myosin head binds. Currently they're exposed, and when they're exposed, the head can bind to them, interact with them, and then perform the power stroke. If these black myosin head binding sites are covered up by the purple tropomyosin cable, then cross-bridge cycling cannot occur, and muscle contraction will not occur as well. So one more time. When these black binding sites on the actin are exposed, that allows interactions with the myosin head and muscle contraction. But when we cover up those black binding sites, the myosin cannot interact with the actin, and therefore we do not get cross-bridge cycling or muscle contraction. As we said on the iPad, this is known as the rigor state, and this is known as the relaxed state. So cross-bridge cycling occurs on both sides of the M line. So this cycle, it occurs at all these different myosin heads, and it occurs when the myosin heads interact with the actin thin filament above it. And you saw that for cross-bridge cycling, it uses ATP. But because cross-bridge cycling occurs at all these different places along multiple sarcomeres, thus muscle contraction will require a lot of ATP or energy. And that's why when you exercise, you need to increase oxygen delivery to your muscles, as well as deliver more nutrients to your muscles in order to make more ATP to power your active muscles during exercise. So with cross-bridge cycling, we'll decrease the length of the sarcomere, the H zone, the I band, and the A band will stay the same way. Okay, so this next section of the lecture will kind of summarize muscle contraction from the top. And when we say from the top, it means from the brain. It's referred to as excitation contraction coupling, where we're having changes in electrical charges from neurons leading to the contraction of the muscle. So this combines the nervous and muscular systems. Where do we initiate muscle contraction? What part of our brain? Or let's start at what lobe of our brain initiates muscle contraction? The frontal lobe. And what's the specific part of the frontal lobe that initiates muscle contraction? The primary motor area. Good. So up here, you'll see a neuron, or an upper motor neuron, that originates in the primary motor area of the frontal lobe. This neuron will then descend down through the brain, cross over to the opposite side, and then extend into the spinal cord. This is one long neuron. And remember, when you initiate muscle contraction, it controls muscles on the opposite side of your body. That's why we get crossing over here. This is the pyramidal tract, which we learned about during the central nervous system. So the signal for muscle contraction begins at the primary motor area of the frontal lobe. And then we'll have a second motor neuron that goes from the spinal cord to the effector. Circled in blue, this is referred to as a neuromuscular junction. Circled in blue is the connection between the axon terminals of this neuron with the muscle fibers or muscle cells. Here's that same axon terminal, and here's the same muscle fiber. They're communicating with each other. And this region, where you have a communication between a neuron and a muscle fiber, is referred to as a neuromuscular junction. It's the area of communication between a neuron and a muscle fiber. So it contains the axon terminal, the synaptic cleft with its neurotransmitters, and the motor end plate. This motor neuron will always release the neurotransmitter acetylcholine onto the muscle fiber. So these purple neurotransmitters are acetylcholine signaling molecules. And if you look at the motor end plate, the plasma membrane of the muscle fiber here, we have these nicotinic receptors in purple, which will bind the acetylcholine. Remember, with nicotinic receptors, when they bind acetylcholine, it will cause depolarization, where a lot of sodium will rush into the cell. So when acetylcholine binds to the nicotinic receptors on the motor end plate of the muscle fiber, it will stimulate sodium ions to enter. And we're going to build up positive charge in this muscle fiber. When we build up positive charge in the cell, recall that we say the cell depolarizes. And eventually that will lead to action potentials that travel down the sarcolemma and then down the transverse tubules, deep into the muscle fiber. So, action potentials. We travel down the sarcolemma and then deep into the muscle fiber using the T, or transverse tubules. Eventually, the action potentials will get to a protein receptor known as DHP receptors. So, if we look closely, let's see. Let's use red. Here's your T tubule membrane. So this DHP receptor resides here. And then you have the plasma membrane of the sarcoplasmic reticulum. Which is located right next door. So you have to imagine that these two structures are located right here. So action potentials travel all the way down the T tubules, and then they'll activate this red DHP receptor. And notice the DHP receptor can communicate with this ryanodine receptor next door. DHP receptors activate the ryanodine receptors of the sarcoplasmic reticulum, and this causes calcium ions to be released. And that's what you're seeing here. Calcium ions are being released into the sarcoplasm of the muscle. Once calcium is released from the sarcoplasmic reticulum, it will then diffuse into the sarcoplasm, and it will interact with troponin. So when the calcium comes out, it binds to the pink troponin, and in a way, it acts as a weight, and it moves this tropomyosin cable in purple off these black binding sites for the myosin head. Because we move tropomyosin off these binding sites, the myosin can now interact with those binding sites, perform power strokes, and allow for cross-bridge cycling and muscle contraction. So that's why calcium is also required for muscle contraction. It exposes the binding sites for the myosin head so that we can get cross-bridge cycling. Calcium binds to troponin. Here, let's color code it for you. Calcium is in green. Calcium ions, they bind to the pink. I don't have pink, so we'll use red. Troponin. And that moves the tropomyosin cable in purple off those black myosin head binding sites. And thus, that allows the myosin heads of the thick filament to then interact with the thin filament above it, allowing for cross-bridge cycling to occur in muscle contraction. So muscle contraction requires calcium as well as, we saw, ATP. In addition, you must get neural signaling to the muscle fiber. So let's talk about muscle relaxation. If our muscles are always contracted, we'll be stuck in a fixed state and essentially we'll be paralyzed. So we need our muscles to relax at the appropriate times. So how do you get a muscle to relax? You kind of do the opposite. First, you need to stop neural signaling to the muscle fiber. You need to stop releasing acetylcholine onto these nicotinic receptors. But you also need to do a few more things as well. You also need to break the cross bridges. You need to stop the myosin heads from interacting with the thin filament above it. How do you break the cross bridge? What molecule do we use? It was on the drawing that we made. What molecule did we use to break this cross bridge? ATP. So here's the cross bridge, and then we said we will bind a new ATP, step number 4, to break that cross bridge or attachment. So does muscle relaxation also require ATP? The answer is yes. How do we break the cross bridge? We use ATP. In addition, we need to prevent the myosin head from interacting with the actin thin filament. When these two can interact, we get cross-bridge cycling and muscle contraction. So we need to stop this interaction during muscle relaxation. How do we do that? Well, we said calcium is required for muscle contraction. So the opposite is said about muscle relaxation. For muscle relaxation, we must remove the calcium. We must pump it back into its storage site, into the sarcoplasmic reticulum. And because we're using a pump, pumps also use ATP. And by putting the calcium back in there, that will then allow the tropomyosin cable to cover those black binding sites. So the myosin and actin cannot interact, and thus we cannot get cross-bridge cycling and muscle contraction. So it's written right here for you. When we get rid of calcium, the tropomyosin will cover the myosin head binding sites on actin. All right, so in lab, we'll talk about how your muscle makes ATP, and we'll finish one of your potential answer questions.